We pop pills, we apply creams, we trust that what's written on the label is what we're actually getting.
Hmm.
But have you ever wondered what's really in our medications? I mean, beyond just those listed ingredients.
It's not something most people think about, I guess, but it's a really good question and actually kind of a big deal when it comes to pharmaceuticals.
Right. Because what we don't see are the unseen molecules that might be lurking even in those carefully manufactured drugs.
Exactly. And that's where a powerful tool comes in, a kind of molecular detective, if you will. And that's mass spectrometry.
So for this deep dive, we're going deep into the world of drug analysis to uncover exactly how mass spectrometry works its magic. We'll be looking at how it dissects the main drug substances themselves and, importantly, uncovers even the tiniest traces of impurities that might be hanging around.
We'll also be talking about the different types of mass spectrometers. Because there isn't just one kind, there are quite a few. And each one is kind of like a specialist, designed for different analytical challenges.
And I guess, like any good detective, a mass spectrometer needs to be sharp. So we'll be figuring out what makes one truly effective.
So we'll be talking about resolution, its ability to tell apart very similar molecules, and sensitivity, its power to detect even minuscule amounts of a
It's
substance.
all about being able to spot the tiniest clues, right?
Exactly. And because sometimes one detective isn't enough, we'll also look at how mass spectrometry partners with other techniques in the lab, like liquid chromatography, or LC for short.
Hmm.
Think of it as a dynamic duo working together to solve the case.
So whether you work in the pharmaceutical field or you just want to understand the science behind what you're taking, or even if you just love diving into the microscopic world, then stick with us, because this deep dive is going to uncover some truly fascinating insights, and we promise to break it down in a way that's clear and engaging.
Absolutely. So let's jump right in. When we analyze drug substances, it's not as simple as just confirming that the main ingredient is there. Mass spectrometry helps us generate a super detailed molecular profile. Think of it like a unique fingerprint for that specific batch of the drug.
So like we can tell not just that it's an apple, but what kind of apple, where it was grown, the whole story.
You got it. And a really important part of this detective work is figuring out what impurities might be lurking within a drug.
Because even a tiny bit of something extra could change things up right.
Absolutely. Even in very small amounts, these impurities, which can come up during the drug's creation or even during storage, they can impact how safe and how effective a drug is.
And I'm guessing that's where mass spectrometry comes in.
Right. It's super sensitive, so we can use it to find even those tiny amounts of potentially risky substances that are hidden within a drug, like searching for a single off-key note in a whole symphony.
You mentioned that there's more than one type of mass spectrometer. Why not just have one do-it-all machine?
Well, the molecular world is vast, and we need to ask a lot of different questions about it. So different types of mass spectrometers have evolved to be specialists in their own right.
So like some are better at analyzing huge complex molecules like proteins, while others are good at finding smaller organic compounds.
Exactly. Some are all about measuring a molecule's mass with incredible accuracy, which is essential for figuring out its exact chemical makeup.
So it's about using the right tool for the job, a bit like choosing between a microscope and a telescope,
Yeah,
depending on what you want to see.
perfectly put. The instrument you choose depends on the complexity of the sample and the level of detail you need. Are we looking for a specific known impurity, or are we trying to identify a mystery byproduct? The answer will often guide us towards a particular type of mass spectrometry.
Now, you often hear the words resolution and sensitivity when talking about these instruments. What do those really mean?
Okay, imagine you have two identical twins, but one has a tiny, tiny birthmark. A high-resolution mass spectrometer is like having superhuman vision that can spot that minuscule difference.
So it's about seeing those really fine details.
Oh, exactly. And in drug analysis, that's crucial. Impurities might have structures and therefore masses that are incredibly close to the actual drug or other harmless compounds. High resolution lets us pinpoint the specific impurity we're looking for and separate it from the rest.
Okay, so resolution is about being able to distinguish between things that are very similar. What about sensitivity?
Think of it like this. You're searching for a single specific grain of sand on a massive beach. A highly sensitive mass spectrometer is like having a detector that can zero in on that one grain of sand, even among millions of others.
So it's about being able to detect even tiny, tiny amounts of something.
And as we said, even trace amounts of impurities can be a big deal. So we need both high resolution to accurately identify what we're looking at and high sensitivity to find it, even when it's in very small quantities.
We've got to be able to see that grain of sand and be sure it's the right one.
Precisely. Now, mass spectrometry isn't always a solo act in the lab. Often it teams up with other techniques, and one powerful partnership is with LC, liquid chromatography.
LCMS, I've seen that a lot. What's the deal with that?
You're right. And in the real world of drug analysis, we're often dealing with a mixture of many different molecules. This is where combining techniques becomes really important. So LCMS is like a dynamic duo that's really valuable in the pharmaceutical industry.
Okay, so the hyphenated just means two different techniques working together.
Yes, basically. Liquid chromatography, or LC, is a separation technique. Remember our different colored candies? Imagine sorting them by color before you try to identify each flavor.
Like lining them all up.
Well, not exactly lining them up, but separating them one after the other. The LC system takes that complex mix of molecules and separates them based on their properties as they travel through a special column.
I see, so it separates them sequentially over time.
Exactly. And then, as each separated component emerges from the LC system, it goes straight into the mass spectrometer. That's where the combined power comes in.
So instead of looking at a jumbled mess of molecules, we get each one individually. Makes sense.
Right, it's much more manageable. Imagine trying to listen to a room full of people talking all at once, compared to hearing each person speak one at a time. LC acts like that filter.
So LC filters it, and the mass spectrometer IDs each molecule.
Mhm.
Makes sense for these complex mixtures. Now, all this data that mass spectrometry produces, it must be pretty complex to interpret, right?
Absolutely. It's not a simple yes or no answer. We get information about the mass-to-charge ratio of different ions, basically a measure of their weight relative to their electrical charge.
Okay.
We also see how much of each ion there is and how they break down into smaller pieces. And we look at chromatograms, which are like visual representations showing the separated components as peaks. All of this takes specialized knowledge to really understand what it all means.
So it's not just plugging it into a computer and getting a result.
You wish. Identifying unknown impurities, for example, and figuring out if they could be a problem that can be really tricky. It involves a lot of detective work. Comparing the data to libraries of known compounds, meticulously analyzing how those molecules break apart, sometimes even using other analytical techniques to confirm what we think we've found.
Sounds like solving a really, really complicated puzzle.
You could say that, and we're not doing it alone. We use sophisticated software and huge databases that contain information about tons of chemical compounds.
Now, in the pharmaceutical industry, regulation is obviously a big deal. You've got to make sure the medications are safe and effective. How does that apply to using mass spectrometry for drug analysis?
It's essential. We can't just use these fancy instruments without proving they actually work and give us reliable results.
Right.
Regulatory bodies like the FDA have really strict guidelines for validating methods in the pharmaceutical industry.
So it's not enough to just have the best tech. You've got to prove it consistently delivers accurate results.
Exactly. We need to demonstrate that the method is specific, accurate, and precise. We need to show it can find the smallest amount of a substance we're interested in and measure it accurately. All of this ensures that the results we get from mass spectrometry are trustworthy and can be used for making important decisions about drug quality and patient safety.
So a lot of hoops to jump through, but for good reason.
Exactly. It's all about making sure those medications are safe.
Now, our sources for this deep dive don't give specific examples of, mass spectrometry swooping in and saving the day, but could you give us a sense of how it might play a crucial role in solving real-world problems in drug development?
Okay, picture this. A pharmaceutical company is doing stability testing on a new drug. They're checking to see how it holds up over time, and they use a technique like LCMS to track it.
Hmm.
And let's say they spot a new peak appearing on their chromatograms. This new peak means a degradation product is forming. Basically, the drug is breaking down into something new. Now, mass spectrometry comes in to identify exactly what that new compound is, like getting its molecular fingerprint.
And knowing that helps them figure out if it's harmful.
Yes. If it is, they can take steps to prevent that breakdown from happening, maybe adjust the formulation, add a stabilizer, or change the packaging, all to ensure the drug stays safe and effective for as long as it should.
So it's like preventive medicine for the medicine itself. What's another example of mass spectrometry in action?
Think about the early stages of drug discovery. Scientists are making all these new potential drug candidates in the lab, and after every step, mass spectrometry acts as a quality control checkpoint. They use it to confirm that they've made what they intended to make and that there aren't any unwanted byproducts or impurities.
So it helps them make sure they're on the right track and not wasting time on something that's not pure.
Exactly. And later on, when they're scaling up production, mass spectrometry helps ensure that each batch of the drug is consistent.
Oh, that makes sense.
If they notice variations between batches, they can use LCMS to compare the impurities. This helps them figure out what might be causing the inconsistency and fix it.
So it's really involved in every step of the way,
Hmm.
from discovery to manufacturing, making sure that the drug is what it's supposed to be. Really interesting stuff.
It is. It's amazing how this tool can help us understand the unseen world of molecules.
So to sum up, mass spectrometry is way more than just a cool piece of equipment. It's like a molecular detective that's essential for making sure that the medications we use are safe and effective.
It helps us see those tiny impurities that can have a big impact, helps us make sure drugs are made consistently, and it even helps discover new drugs.
And it often teams up with other techniques like LC to get the job done.
Exactly. We talked about how important resolution and sensitivity are and how the whole process is tightly regulated to ensure accuracy and reliability.
It's amazing to think that we have this level of molecular detail available to us, isn't it? It makes you wonder what other areas of our life could benefit from this kind of deep dive, this close look at the molecular level. What other hidden worlds are out there?
It's a great question, and it really highlights how important analytical chemistry is. It helps us unlock the secrets of the world around us, piece by tiny molecule.
Thanks for joining us for this deep dive into the world of mass spectrometry in drug analysis. Until next time.
Thanks for having me.
Bye.